our final speaker before the break. We have Hang-Ying Yang from Pennsylvania State University.
Thank you. So today I'm talking about my work on the cathode-influenced metabolic flux distribution in exoelectrotrophic Clostridia. So the species I'm focusing today is the Clostridium acetobutylicum. This is a very well-known anaerobic bacterial. It is gram-positive and has been intensively studied in the past century for the biobutanol fermentation, which has recently been proposed for alternatives to the fossil fuels as the fuels for the transportation. The Clostridium acetobutylicum has both a cetogenic process and the solventogenic process. This is the original figure showed the metabolic pathway, but it's too small, so I made a simplified figure here. With the glucose as the substrate, the Clostridium acetobutylicum can first have the acetogenic process for the production of the lactate, acetate, and acetone and butyrate. When there is accumulated acids, end products and the pH lowered to lower than 5.5, it is switched to the solventogenic process for the production of alcohols such as ethanol and butanol. The production of the alcohols, the ethanol, and the butanol requires more electrons from the acids here. The production of the butanol from the glucose is usually at a high feedstock requirement. And there is a previous study showed that the carbon and electron flows can be altered by electron flow modulators. So they used a mediator, methylviolagine, as the mediator, and set the potential at very low value. And they found that the production of the butanol to acetone ratio increased from around 2 to 3.3. And recently, Clostridium acetobutylicum has been demonstrated to be able to generate current in MFCs without the addition of redox mediators. Based on very high glucose concentration fed in one batch, they actually found 2 peaks of the cell voltage. The first one is correlated to the production of the hydrogen and the acids. And the second one corresponded to the switch from the acetogenic process to the solventogenic process. So in my research, my first hypothesis, the Clostridium acetobutylicum can take up current without the addition of mediators, which means that it's exoelectrotrophic. And the second one, if this species is exoelectrotrophic, I'm wondering if the electrons supplemented from the cathode will affect the biosynthesis of the organic products, also the hydrogen production by this species. So I adopted the 2-chamber reactors with nephium as a membrane, and I fed the cathode with the pure water, and the anode with the pure water, and the cathode with the YTG medium contains the tryptophan yeast extract, sodium chloride, and glucose, and inoculated with the ADCC824. I used glucose here because this strain does not have the Wood-Linda pathway for the carbon dioxide fixation for autotrophic growth. The cathode potential was set around -400 millivolts versus standard hydrogen. And for around 2 months running of the reactors, we have found several batches of the current uptake in the reactors. Before we inoculate the reactors, we didn't find any hydrogen production at this cathode potential for 1 day. We achieved up to 83 milliamps per square meter current density on the graphite plate cathode. However, we have also found a decreased trend of the current uptake through each batch, although we have fed them with a fixed glucose concentration for 1.5 months. At the end of this 2 months, I've run my reactors first in the closed circuit as previously and also left them in the open circuit as a batch to compare the difference. I found that the hydrogen production reached to around 30% of the total gas composition in the cathode head space. However, there is a difference of the hydrogen concentration at the later of the batch, which showed a decrease trend in the open circuit, but not detected in the closed circuit. Another thing we detected is that before the open circuit operation, the two duplicates are required like similarly or respond similarly, but after the open circuit, one of the biofilm just failed to take up current again. So I set up another experiment, separate the closed and open circuit group to avoid the differences along the time courses, because you see the difference of the current uptake during the batches. So the group A was set as closed circuit for the 4 batches, and the group B set exactly the same for the 2 cycles, first 2 cycles, and then left as open cycle for the last 2 for comparison. In order to minimize the effect from the suspended growth to the effect, we have lowered the glucose concentration. This is around like 1 gram. These are 12 millimolar, and then down to half of it. And then we also transferred the cathode electrode to clean cathode chamber during batch 3 and batch 4 to also prevent the growth on the cells grow on the wall of the reactors. The L-cysteine was added as oxygen scavenger because we wanted to exclude the oxygen reduction happen in our reactors. We also use the same reactor type, except that we used the YTG media without the glucose in the anode to enhance the conductivity. So we found that first we found a similar trend of the current uptake as we see before. And in the first 2 cycles, when group A and B both in the closed circuit, they showed a similar current uptake, which showed that they were comparable. And in the last 2 cycles, only the group A were set as closed circuit. The current uptake showed the mysterious decrease as previously, and the glucose quantified by the sulfuric method showed around 60% consumption during each batch. The CV test was done on the abiotic reactors before the inoculation of the Clostridia acetobutylicum, all exactly the same feeding on the cathode and anode. And there is no mediators in the influent. And also, CV was done on the end of the cycle 4 on the catholyte with suspended cells, and cathode. Cathode is the red one, and the catholyte is the blue one. And we can see that there is no mediators in either the medium or the effluent, but the biofilms showed a response at the potential, showed a response of the current uptake at the potential that we set. This demonstrated that the biofilm of Clostridia acetobutylicum is exoelectrotrophic. The significant hydrogen production usually occurs on the 2nd day of each batch, and before the cycle 3, the group A and B showed similar performance. However, when the group B was left in open circuit, there is an oxygen oxidation phenomenon showed in the group B, but this was not found in the closed circuit of group A, and also when the group B in closed circuit before. At the end of the 4th cycle, the hydrogen production, more hydrogen production per mole of glucose consumed is higher in closed circuit group A than the open circuit. Very interesting thing is that although the cycle 1 and 2 were fed with the same levels of glucose concentration, there is a similar trend of the decrease of hydrogen, decrease of trend of the hydrogen production as we see in the current uptake decrease. Probably they were related. The conversion of the acetyl-CoA to butanol requires more electrons than the butyrate, as I've shown you in the previous slides. Here we have seen increased butanol production in the closed circuit, while decreased butyrate production in the closed circuit. We have also seen a similar trend of the decrease of butanol production from the cycle 1 to 2, similar to the current decrease of the current uptake. Although when you first look at this figure, it looks like that the decreased butyrate contributed to the increased butanol, but I have to mention you that they have three different magnitude difference of the concentration. The ethanol production from the acetyl-CoA also requires electrons, but the conversion of the acetyl-CoA from to acetone actually does not involve electron consumption. Here we didn't see too much difference of the acetone production for the closed and open circuit, but we do find a decrease of the ethanol production in the closed circuit. Actually, a previous literature has showed that when there are more electrons available from either the cathode with mediators or more hydrogen there might be a switch from the acetate to ethanol by consuming more electrons. However, the acetate concentration here, we tested it, but we cannot quantify it because we have a complex background in the medium, and we are encountering a background interfering in HPLC test during the peak of the acetate. The pH change showed that in the first 2 cycles, during the because of the acid production, the pH dropped to an area that the acetogenic process can be switched to solventogenic processes. However, with lowered substrate level and lower acid production, the pH drop in the last 2 cycles were kind of like the edge of the switch from acetogenic to solventogenic. So as a summary of the change of the metabolic flux with the current uptake, we have found increased production of hydrogen and butanol here, which all requires electron consumption. But we found decreased ethanol and butyrate production. The acetone production, which does not involve electron consumption from acetyl-CoA, did not see too much difference. And unfortunately, we cannot quantify the acetate concentration here to give a closed electron balance here. So as a conclusion, first we found that the Clostridia acetobutylicum is exoelectrotrophic with a need for the supplementation with an organic carbon source. However, the magnitude of the current uptake showed a decreased trend along batches for unknown reason. The cathodic electron delivery resulted in increased hydrogen and butanol production, but decreased butyrate and ethanol production. The acetone production was not significantly affected. The acetate concentration was tested, but could not be quantified here due to background interfering, but this might also related with the electron flow balance in the metabolic pathway. So I would like to thank my advisor, Jay Regan, and the lab, Dr. Logan and the lab, and specifically I would like to thank Shuping and Dave for helping me solve the problems and on the GC and HPLC. The research was supported by the Quest and Army Research Office Equipment Grant. And thank you very much.
Are there any questions?
I was wondering, your current decreases over time, can that relate to scaling on the cathode?
Scaling? You mean the size of the cathode?
Scaling in a way that you have a low pH increase at the cathode, leading, if you have quite some calcium in your medium, to calcium carbonate precipitates, and that can kind of passivate the electrode.
Yeah, that's possible because I've seen like biofilm growth on the cathode. Yeah.
I noted that you add yeast extract to your cathode light, and I'm always worried about that could act as a mediator. But you show some CVs.
Yeah.
That's most likely not. Have you thought about it? Have you done some more experiments?
Yeah, we also heard that there is publication showing that yeast extract probably can serve as like a substrate for the growth of those anaerobic cultures. But that's why we did the CVs on the medium only before the inoculation. But based on what we've seen here, we didn't see that because we have significant different magnitude of the current uptake when we inoculate the Clostridia before compared to without. So we don't think that that will cause a problem to the current uptake. Yeah.
Would you like to join me in thanking all the speakers, Hangjing and all the speakers from this session?
Thank you.